right, I'm going to introduce our speaker. I'm Dr. Youssef. I'm with the Division of Nephrology and Hypertension. Our speaker today is Dr. Michael Merchant. He did his training at the University of Arkansas. And actually, I didn't realize this, but you've been here since 2003. 2003, presently a professor at the school at the University of UofL. Dr. Merchant does many things. He is the co-director of the omics facility, both for the school and for the alcohol center and the hepatology center. But remember, he's working in the division of nephrology, so I know he's got that kidney stuff there somewhere. He has over 130 publications. He is a reviewer for multiple journals. And today's talk is the spatial proteomic studies of archived formalin-fixed paraffin-embedded tissues and the applications beyond the kidney. Thank you, Mike.
Thank you, Rosemary. So I want to thank all of you for showing up today. I want to thank Dr. Williams for making this all possible as well. It's kind of a very challenging thing to follow up many of all the talks this year. They're all very mechanistic. Coming from proteomics, there's a lot of observational things that we build into a hypothesis. I think that part of the goal for this talk is to establish kind of translational bridges between research and clinical studies, clinicians. So I'm going to stick with the theme of some discovery science in my talks today. So I have disclosures of where we have funding from, both federal and from the kidney disease program. Basic learning objectives are to address the approaches that we use to study microproteomes, to review some glomerular disease, and then discuss the application of the methods to spatial proteomics. As an outline, I want to cover a little bit of a historical perspective for how we understand renal disease, jump into rare diseases, discussions of what a rare disease is, kind of acknowledge some of the challenges that exist because of that. Discuss innovations in proteomics that are being used to address spatial omics. Cover a little bit of the matrixome because I'm interested in the extracellular proteome and how that guides cell fate. The specific spatial proteomic applications that I'll address are all renal disease, but some recent collaborations with Igor and Sanjay on some cardiovascular disease projects. Historically, kidney disease developed out of a field of science, pseudoscience called uroscopy, or water casting, where urine was collected in a flask and the color of the urine was compared against a color wheel that somehow was used to diagnose disease. There was also the old smell and taste tests as well. So we've come a little bit of a longer, we've come farther than that. So historically, the field of modern pathology, Western renal pathology, might be considered to be initiated in the 1660s with the development of the first microscope. And then the progress through the innovations contributed to the different individuals in the 1800s and 1900s, where the field led up to using a renal biopsy method to retrieve tissue from an individual percutaneously, and then characterizing that biopsy using a variety of histologic methods and electron microscopy methods, leading to the diagnosis of disease. So you might say that this took some 300 years to get up to the stage of having some molecular information. Following that development of the immunofluorescent tools, there's been a fairly rapid development, comparatively speaking, for the application of molecular tools for the identification of renal diseases. For instance, immune complex formation was discovered in the 60s for lupus, IgA nephropathy, post-infectious nephropathy, development of information for focal segmental glomerular sclerosis was in the 70s. ANCA disease, anti-neutrophil cytoplasmic antibody disease in the 80s. Membranous nephropathy, another glomerular autoimmune disease in the 80s. Hemolytic uremic syndrome, the 90s, podocyte disease, transplant disease in the 90s. So progress sometimes takes a long time to evolve and then rapidly has a punctuated jump into molecular findings. So mechanisms always seem to stem from identification of associations leading to causation. So to establish kind of where we are in the realm of impact on medical science on patient care, if we look at the prevalence of chronic kidney disease, within the US population, we see that there is a bias for age, older adults having higher prevalence of kidney disease. There's a small bias for more kidney disease in women, and a bias for kidney disease being observed in non-Hispanic Blacks. So everything is not cut and dried. There's not a common cause for everything. There's a nuance across all these fields. And so we are challenged with our discovery science to assemble cohorts of patient samples and develop approaches that allow us to uniformly examine those samples and those tissues for comparative purposes. Within that spectrum of etiologies for kidney disease, reported causes for end-stage renal disease are biased towards metabolic diseases, so diabetes, bias for high blood pressure, obesity associated, and then the less prevalent glomerular nephritis causes and the other unknown secondary causes of chronic kidney disease leading to end-stage renal disease. So one of the rare diseases that's actually not a disease is nephrotic syndrome. This is a renal disease, if you will, that's characterized by massive proteinuria, edema, and hypoalbuminemia. This affects children and it affects adults. And causes for nephrotic syndrome are minimal change disease, focal segmental glomerular sclerosis, membranous nephropathy, IgA nephropathy, and diabetic nephropathy. So what am I trying to emphasize here? I'm trying to emphasize that oftentimes renal diseases are captured at a primary care visit with the collection of a urine sample or from patient characteristics of edema. There is no clear-cut observation of a viral load associated with high temperature during the COVID era that you get tested for and you identify kidney disease. So it's a very nuanced thing. When it presents and is detected, it may appear at a variety of stages in the progression of the disease. So not only do we need biomarkers that efficiently detect and diagnose the disease, but they need to be applicable to race, sex, age, as well as stage of progression. So it's a challenging subject. Another challenge for us is that the rare diseases, not the diabetic nephropathy, which occurs in millions of people globally, are the diseases such as focal segmental glomerular sclerosis. That's a rare disease. So NIH classifies a rare disease as a condition that affects fewer than 200,000 people. So with it being a rare disease, the challenge is that not everybody is seen in a timely manner by the physicians that might be located in major centers that would allow for recruitment of those individuals into clinical studies. So from the perspective of translational research, being able to access large numbers of samples or patient specimens to efficiently and sensitively identify biomarkers is a challenge. So on our end, we try to do things that can maximize the information we can obtain from what's available to us. Oops. Okay, so I want to highlight a couple of publications that we've been part of that highlight a punctuated jump in the field of patient care in kidney disease, specifically in glomerular disease. So collaborations with Boston University have led to the identification of 2 different target autoantigens for a type of rare disease called membranous nephropathy. So membranous nephropathy is an autoimmune disease where you have subepithelial basement membrane deposits of an antibody immune complex that leads to a variety of things, ultimately leading to glomerular sclerosis and failure of the kidney. So these studies, the senior author here, David Salon, the first author, Larry Beck, David Salon pursued this line of investigation for 40 years, hammering away at a rare disease, and a chance interaction between Larry with John Klein and between David and John Klein and Larry Beck and Dave Powell led to the early stage use of proteomics for identification of proteins in a gel band. And Tim Cummins, our faculty member, ran the samples on the mass spec, and they returned a list of proteins in that gel band. And Boston University investigators hammered away using antibodies for these various proteins. And they identified one protein, the M-type phospholipase A2 receptor, as being the autoantigen responsible for 70 to 80% of this disease. So this has led to diagnostics for this disease. It's led to therapeutics. It's been led to the ability to stage the response of individuals for various therapeutics. It's led to a lot of progress in the field. So 2 things I want to point out here is the application of spatially refining your question and the use of discovery science to build up an inventory of targets that you then individually refine. So it's going to take a team for whatever we do. So in what they did, they isolated, they minced kidney cortex from a donor kidney. They isolated glomeruli, the microvascular bed. They eluted out of those glomeruli the antibodies, and then began to use those antibodies to determine by immunoblot the presence of the autoantigen, and then cutting out those gel bands. That's what was analyzed by proteomics. Other rare diseases that fall within the arena of nephrotic syndrome have a poor diagnostic classification. So I mentioned previously that diabetic nephropathy is one of the conditions that can lead to nephrotic syndrome. So keep in mind that when a patient presents with nephrotic syndrome, the typical approach for identification of the etiology is a biopsy. So somebody's going to lay on a table, have a needle jammed into them and tissue taken out. And then histologically, that's going to be evaluated to determine what class of disease it is. And if the markers for that class do not present, then you're left with an unknown. So even though some of these rare diseases impact only 10% or 5% of diabetic nephropathy, at the start of the whole process, it's a complete unknown. So you have to test for everything. So anything that you don't test for, that remains an unknown. So IgA nephropathy is diagnosed on biopsy by IgA immunofluorescence. And so that kind of classifies that disease and allows the clinicians to address the patient care appropriately. Membranous nephropathy these days is diagnostically identified by the autoantibodies to a range of known proteins that correlate to about 90% of the patients with membranous nephropathy. Diabetic nephropathy, you all know that very well. You're looking for hemoglobin A1C, you're looking for fasting glucose levels, you're looking for insulin levels. There's a variety of tests for diabetic nephropathy. So that's not really the challenge. Minimal change disease and focal segmental glomerular sclerosis remain the pieces of the pie for nephrotic syndrome that are a challenge. So we particularly wanted to address in our application of laser capture, microdissection, and proteomics, to renal disease, nephrotic syndrome studies, we wanted to focus on focal segmental glomerular sclerosis, or FSGS. So FSGS is somewhat of an unknown entity. Histologically, it can be characterized by the type of sclerosis patterning in the microvascular bed. But there are primary forms of FSGS, which is basically an idiopathic form, an unknown form. And then there are secondary causes of FSGS. So the patterning of sclerosis can be associated in a secondary fashion if somebody has certain types of hypertension, obesity, certain types of infection, viral infection, parasitic infections. Drugs can drive that formation of sclerosis. So there's a range of causalities associated with the phenotype. In the absence of knowing which one of those etiologies which one of those secondary causes drives it, you're left with a little bit of a question on how to intervene, how to treat. So we sought to fill in some gaps with the idea of developing better surrogate diagnostic approaches. And hopefully those things that we identified might be part of a disease mechanism. So that might lead to target engagement biomarkers, entities that there might be a known drug on the market to modify its activity. So we're going to fill in the gaps with our omic approaches, looking at spatially resolved samples, and we're going to be sensitive to changes in the cellular versus the extracellular proteome. So some of you in the room operate CyTOF instruments. You know a lot about this method. So I'm not going to dig into this very much because I don't know much about it. But it is a methodology for spatially characterizing proteins in tissues. And you're spatially characterizing those proteins because you're using antibodies specific to your target protein, and you're able to mix a range of antibodies, anywhere from 40 to 100, I think, antibodies into your sample. And then using mass spectrometry, you can localize and in a relative fashion quantify the amount of that antibody present. And you're able to build up some sort of image representation that you could then overlay on some histologic image and identify abundances of proteins associated with structures in your tissue. So that is a type of spatial omics that has been really enabling a lot of research locally and nationally. Another type of spatial omics is a MALDI-based imaging analysis. And with this approach, you're basically taking a tissue section from a frozen state and you're sectioning it just like you would for histology. You have to apply an organic compound to the tissue. The organic compound, as it dries out, forms a crystalline matrix. And then a laser is fired at the tissue in a very small region, 2 microns, 3 micron area. And that laser vaporizes the matrix, and anything that's dissolved into that crystalline matrix also is vaporized. And so you can collect mass spec data on the plume of ions that comes up out of that. So this is an example of image analysis by MALDI of a tissue section from somebody with membranous nephropathy. And so on the left-hand side, there are 2 different blue stripes, I guess. That's the tissue of the biopsy. Those colors are all pseudocolors based upon the intensity of some ion. So every MALDI image is a pseudocolored image where you're extracting a channel, a mass spec channel of information, and you're ascribing a color to the total intensity or area of that signal. And so you're having a pseudo representation of ion abundance, and you're inferring something from that based upon your knowledge of what that ion is associated with. If it's a lipid, then you're studying spatial lipidomics. If it's a protein, then you're studying spatial proteomics. If it's a glycan, you're studying spatial glycomics. So the left-hand side is the difference in those 2 images is just the resolution of the laser that you're using to fire to volatilize your matrix. So the smaller the laser beam you have, the better resolution in your image, the better you're able to capture that information for the spatial representation of your omics. And so on the right-hand side of my screen, right-hand side, you see that there's the capacity to overlay images from the MALDI with PAS stained sections. And so these come from overlaying serial sections. So you have one section that's stained with PAS, another section that you capture image from CyTOF or MALDI, you collect your channel of information, you overlay it, and you begin to extrapolate the presence or absence of some species with the histologic information, such as fibrosis. And then you can perform on-tissue digestion and convert the proteins into peptides and collect a little bit higher resolution data for proteomics studies. So this is just one representation for a fragment ion of a sonic hedgehog and collecting data spatially between the glomerular compartment and the tubular compartment of the kidney. So I didn't go through this because it's another slide and I've already got 50-something slides. So you're going to be throwing stuff at me already. But the functional unit of the kidney is the nephron, right? So you have a microvascular bed that ultrafilters the blood. The blood ultrafiltrate comes into the urine space and is funneled through a tubular cell network down to through the renal pelvis into the bladder. And so every step along the way through the different segments of the nephron, those epithelial cells have specialized processes for recapture of certain types of analytes. So there are a lot of channels throughout that network for protein, for salt, for water, for sugar, et cetera. So this is just one way for us to drive into collecting spatially refined proteomic data sets. The other way to develop a spatially refined proteomic data set is to do micro or nano proteomics. And so in this approach, you're either going to isolate a very small piece of tissue, which is what we're going to do with laser capture microdissection, but you can also use flow cytometry to isolate cells. And then you're carrying out your entire proteomic workflow within a small volume that's called a pot, a POT. So you have micropots where you develop using everything in microliters or nanopots where it's in nanoliters. And so people have demonstrated the capacity to analyze individual cells at this point in time. This is from 2018. Currently, people are performing single cell proteomics with specialized instrumentation. So yeah, I need to talk fast. So the hypothesis for my research actually came about from discussions with other people on campus. So back in around 2012, 2013, I was recruited into the Alcohol Research Center by Craig McLean. So I need to give him props for this. I interacted with Jesse Roman, who was looking at lung fibrosis as associated with nicotine exposure, alcohol exposure. He was working with Gavin Artiel, correspondingly in the liver. Both of these individuals were studying transitional remodeling of the ECM, which I thought was an incredibly sharp thing to think about. The basic concept is if you have you know stories about people who experience something like lung cancer, although they have never smoked. And so the thought is that they haven't done anything intentional to increase their risk for cancer. Or you hear people that rapidly develop some tumor. Sorry, Levi's going to kill me. But they rapidly develop metastatic disease. And so why does the course of the disease occur so fast and other people it's so slow? So this is similar to the kind of the seed in the soil type of theory with cancer. So Jesse and Gavin both independently and collaboratively have demonstrated in lung and liver exposure to nicotine or exposure to alcohol can enhance expression of extracellular matrix proteins such as fibronectin or collagen 1A1, but histologically these tissues look identical. There is no difference. There's increased expression of these extracellular matrix proteins, but histologically they look the same. So the challenge for us in proteomics would be we have to be guided by some information to define the sample sets that we analyze. So in the absence of any knowledge, we would probably pull these samples together and the data would be signal averaged and we would have lower quality data sets. So Christine Dolan was a jointly mentored graduate student. She's long since gone and been board certified in clinical chemistry, but she worked for Gavin and myself in liver and in kidney using kind of a sequential extraction process for taking, in this case, liver from mice that have been exposed to ethanol or exposed to LPS or a mixture of the two. So a kind of a one-hit, two-hit type of hypothesis. And in her sequential extraction process, she took the liver and extracted it with sodium chloride. And so that's going to just extract out the freely soluble proteins. She then hit it with SDS, which is going to disrupt membranes, pull out another tissue compartment. Then hit it with guanidinium hydrochloride, which is a very strong chemical denaturant. So we're going to help solubilize some proteins out of protein complexes, high molecular weight complexes that may have not have dissolved into SDS. And then we're left with a pellet. And the pellet itself is digestible. And so with proteomic analysis, we were able to demonstrate that proteins can appear in multiple compartments, multiple extraction compartments. So the thought process is that these represent proteins either in different stages of expression and secretion, so cytoplasmic versus extracellular or nuclear, or they represent proteins that are present in a very tightly bound complex, maybe perhaps cross-linked to the extracellular matrix, and would represent a different ECM phenotype. So this is just an example of plasminogen, an XNA1, or fibrinogen gamma chain, and the presence across a variety of extraction domains and different treatment conditions. So this supports the idea of a transitional matrix, extracellular matrix. I'm going to go through this pretty fast. I think a lot of you in the room do research on the ECM, and you look at it in your various organ states. So ECM is no longer collagen-centric or fibrocentric. When we talk about ECM or extracellular matrix, we're not talking strictly about structure. We're actually talking about a variety of proteins or biomolecules that modify that compartment. So the ECM can function as an adhesive substrate for cells to crawl along. And when that substrate is modified, it affects chemotaxis. It provides structure, structural rigidity. It actually acts as a repository for growth factors to the receptors. It can sequester and store those growth factors as proforms, and it can sense and transduce mechanical signals. So the kidney is consistent with everything that I've just said. The kidney has the interstitial matrix comprised of a few well-known structural elements, but it has a variety of functional components that our goal is to discover. And the interstitial matrix of the kidney functions mainly as a tissue scaffold. We have a basement membrane comprised of collagen-4, laminins, perlicans, and it anchors both the epithelium and the endothelium. And then we specifically have the glomerular basement membrane, the basement membrane of the microvascular bed, that functions as an ultrafiltration device. So that basement membrane has a fenestrated endothelium on one side and a podocyte layer on the other side. And it's a little bit more freely soluble for lower molecular weight species than the other basement membranes, such as the tubular compartment. One characteristic of that basement membrane is it has to withstand the filtration forces. So it has to transduce signals for pressure, but also maintain support to withstand that filtration support. I want to skip the left side. I'll mention, well, not skip it completely. The matrosome was a concept developed by Alexandra Nava when she was at Harvard. She's now at University of Illinois, Chicago. And this is the concept that defines the 5 different compartments that comprise the matrosome: the secretive factors, the ECM affiliated, the regulating factors such as proteases and protease inhibitors, and then the glycoproteins, collagens, and proteoglycans. We see normally with aging that there are changes that take place in the ECM that are consistent with the heart and liver, so consistent with other organs. Transitional remodeling of the glomerular ECM in rare kidney diseases is where I chose to apply my experiences with Gavin and Jesse. So within the field of glomerular disease, I focused on FSGS. FSGS, there's a primary component, which is idiopathic, which means it's a complete unknown. We have viral associated FSGS, so viral associated being associated with HIV and COVID-19. We have FSGS associated with medications and toxins, adaptive FSGS, such as with hypertension, obesity. We have APOL1 associated FSGS. This is a new field that's developed within the past 10 years. I'm not going to really address that other than to say it specifically impacts individuals of West African descent. And then there are genetic components to FSGS. So as I mentioned earlier, everything that the primary method for identification and diagnosis of some type of etiology of glomerular disease starts with histology. And so you can see that most of these stains are Jones or silver stains. You see in the middle the microvascular bed of the glomerulus. Surrounding it is a parietal epithelial cell layer that forms Bowman's capsules. And then surrounding that, you'll see a variety of circles that are the tubular compartment, either proximal or distal tubular compartment. In some cases, you can see that the glomerulus seems to be attached on one side to a rather large structure. This is the vascular pole of the glomerulus, and at the other end is called the urinary tip. So that's where at the other side the nephron, the proximal tubular portion of the nephron, leads away and conducts urine ultimately to the bladder. So these staining patterns, this staining pattern in particular, looks pretty much identical to that of a normal individual, somebody without kidney disease, somebody without glomerular disease. There's no difference in the shape of the staining of the silver stain. There's no concentration of stain in a particular area that might indicate sclerosis. You have a lot of openness to these structures that show the capillaries are open. So then you have, in others, you have characteristic areas of staining that take place. In this case, it's at the urinary tip. So this is called the tip lesion variant. We have some staining patterns that take place near the urinary pole. So this is called the perihilar variant. Some staining patterns are not specific to any specific pattern. So it's called not otherwise specified, or NOS. And then you have a variant where you have a centralized collapse and replacement of the glomerular tuft with extracellular matrix, and this is called the collapsing variant. So the significant aspect of the collapsing variant is that when biopsy is taken, the guidelines or the hope by the renal pathologist is that they'll see at least 10 to 12 glomeruli within their field of view. And if they observe one glomerulus with a staining pattern consistent with the term collapsing variant, then that will be the diagnosis. So just 1 out of 12 is sufficient. And that has been, through the course of various case studies, identified as having the poorest prognosis for response to any therapy and the most rapid progression to end-stage kidney disease. So we selected, we hypothesized that considering the extracellular matrix of these various forms of FSGS, that there existed a transitional nature to the extracellular matrix that guided the cellular activity that led to the extracellular matrix accumulation. Since the patterning of the NOS was not specific to any general pattern, we selected NOS versus FSGS as our phenotypic extremes to characterize. And so our general workflow is to work with clinicians and pathologists to assemble our sample sets. In this case, we're using biopsy material that we then isolate with laser capture microdissection. We profile it with proteomics, and we begin to develop some hypotheses about surrogate markers of disease activity. So everything is mass spec based in our workflow. We had some background information, some studies in the literature that predicted our ability to characterize the proteome of the renal glomerulus. This was a study, one example of a study conducted by somebody at the University of Manchester, Rachel Lennon, who is phenomenally productive in this field. In this instance, much like the study I mentioned earlier with Larry Beck and David Salon, they isolated human glomeruli. They then extracted the glomerulus with the sequential extraction technique that we used on the liver. And they took those fractions and ran them out on the gel and cut gel bands and performed mass spec to identify variations in the extracted fractions for composition. And they were able to identify enrichment of extracellular matrix proteins in the glomerulus that are different in relative abundance for the rest of the kidney. So that led to some information on specificity for composition. Our approach was a little bit different in that Michelle Barati is able to take tissue sections and on glass slides or on PET membranes and place them on the stage of this particular microscope, and she's able to capture an image, a light image of that, and then is able to use the software to define boundaries within the tissue that we would like to have segmented by a laser that's fired through an objective. So that laser then follows that pathway that's been defined in the software and is able to essentially cut out that region of the tissue, which drops into a cap, and then we use that as our sample set for proteomic studies. So this would be an example of tissue from a common source that was first, some of which was fixed and embedded in paraffin, and some of this is frozen. So this is the before and this is the after from retrieving the glomeruli, and that's what the glomerular section looks like in the sample cap. Typically, we've been doing this with 10, 5, and 3 micron thick sections. So you're able to calculate the area of the circle that you define, or whatever irregular shape. The software will calculate an area, and then based upon the thickness, you can calculate a volume, and you can estimate numbers of cells. And in the 3 micron thick sections that we're studying for glomeruli, we're coming in around somewhere around 3 to 10 cells per sample. So we conducted a proof of principle study using the frozen and the fixed samples from normal individuals, 3 normal individuals, or 3 individuals who we had samples from that were not identified as having a known kidney disease. Nobody gets a biopsy unless there's a reason, right? So we performed a sequential extraction approach on our glomerular sections. And we characterized the extracellular matrix fraction to what Rachel had published. And we saw a good overlap, but we saw consistent differences with what she had published. Okay, so I'm going to jump ahead because that's not important. So we then translated that methodology into the study of collapsing focal segmental glomerular sclerosis versus the not otherwise specified FSGS. And in this case, we had available to us 7 patient samples, 7 biopsies. This is a rare disease, and our ability to network out to renal pathology labs and sign MTAs that provide tissue is difficult. So this kind of limited our study, and we had a balanced number of FSGS NOS samples. But the 7th one that should have been here, subsequent paperwork we received identified a different kidney etiology that led us to exclude that. So the tissue was retrieved by laser capture by Michelle Barati. Then people in the lab extracted. Danny Wilkie performed a differential extraction that allowed us to partition the cytoplasmic versus the extracellular component. We studied it with some proteomics. We collected the data and carried it through some different types of analyses that allowed us to interpret our data. So the proteomic data that we retrieved for these samples, we first analyzed by kind of a PCA-like type method to basically ask the question, is the proteome in the one group of samples different from the other group? So this is just giving you a generic characterization of global differences. And what we saw was that in the first dimension, the dimension that explains the largest amount of variation in the data, that separated all of our collapsing from all of our NOS samples. When we looked at our differential abundance between our two groups with a volcano plot, what you can see is that the samples are strikingly different. I'm just going to approximate about 400 proteins differentially abundant out of about 1,200. So about 200 increased in collapsing, 200 decreased in collapsing. We looked at these by filtering for the extracellular matrix proteins using a database developed by Alexandra Nava called the Matrixome. This is a representation of those proteins that were filtered out by a volcano plot. Then looking at the proteins based on their ability to form predicted protein-protein interaction networks using StringDB, allowed us to dig into the groups of proteins and classify them according to the matrixome itself. So we have a group of regulated proteins that are proteases. We have protease and protease inhibitors. We have cell matrix proteins, and we have collagens. And then we have the ones that were unknown, right? And so we looked at a variety of pieces of information that we had available to us to identify what proteins we wanted to follow up for confirmation and validation. We selected 3 out of this set, cathepsin C, cathepsin B, and annexin A3 for validation. And in fact, we chose about 20, and Michelle slogged through all of those. And the differential extraction approach gave us some false positives for having an extracellular matrix component. Some of the antibodies stained heavily on the cytoplasmic side. So we focused on those that stained on the extracellular compartment. We tested those antibodies against biopsies from all the other different FSGS etiologies, as well as disease comparisons for minimal change disease, IgA nephropathy, membranous nephropathy. And we saw consistently with these 2 proteins a specific expressional pattern present in the gloms of the patients with collapsing that led us to then troll through the literature and find other data sets, transcriptomic data sets, where people had compared FSGS versus other control and disease states by using microdissection of patient biopsies, extracting the RNA and performing RNA-seq or RT-PCR. Most all of them were RNA-seq based. And what we saw was that consistently these 2 proteins were upregulated in FSGS, and in some cases specifically demonstrated to be upregulated in collapsing FSGS. So we looked to examine whether or not these proteins were co-localized to a common cell, because our hypothesis of the seed in the soil and the extracellular matrix being different in this etiology and perhaps recruiting or activating uniquely subpopulation of cells led us to the need to be able to demonstrate co-localization. So we were able to co-localize annexin A3 and cathepsin C and cathepsin B all together into common cell types or common cell space. We also were able to co-localize annexin A3 to a protein claudin 1, which is a marker protein for the parietal epithelial cell. It did not co-localize with the marker for the podocyte, and that's significant because the world of nephrotic syndrome and glomerular disease is podocentric. Everybody considers podocyte to be the gatekeeper of disease. And this is demonstrating that collapsing FSGS, that unique variant that has a poor prognosis, is perhaps not a podocentric disease. It may be parietal cell centric. So the parietal cells are an epithelial cell type that line Bowman's capsule. They extend up into the vascular pole, and there is data to suggest that these cells can de-differentiate and transdifferentiate back into podocytes or other proximal tubular cell types. This is an electron micrograph of a parietal epithelial cell with a primary cilia extending down to interact with podocytes. So many glomerular diseases result in the shedding of podocytes. The podocytes lose their foot processes that allow them to grasp around the capillary. They lose those foot processes and they release from the basement membrane and are lost in urine. And that denuded basement membrane has a certain amount of reactivity to it. And so this is a mechanism that is thought to be one possibility for how a parietal epithelial cell is able to sense a denuded basement membrane, become activated, and actually migrate across urine space and bind to the basement membrane and de-differentiate and redifferentiate. So we did some expanded histologic studies where we looked at the staining pattern, the area, or simply the fraction of glomeruli that stained with our marker proteins, and took those values and performed a receiver operator characteristic curve analysis to look for the ability of our marker proteins to differentiate collapsing FSGS from everything else. And what we found was that whether or not we looked at the fraction of glomeruli staining or the area, we achieved a really sensitive and specific detection for these biopsies when blinded. And the significance of these findings is that the approximately 50% of the glomeruli are being stained for these marker proteins in these patients, as opposed to just the collapsing sclerotic phenotype is only in about 1 out of 10 or 1 out of 12. So it's a more sensitive diagnostic method. We have jumped into the field of viral associated FSGS to see if this is consistent with a kind of a central unified field theory for collapsing phenotypes. So HIV associated nephropathy is well known to be associated with acute kidney injury as well as collapsing glomerulopathy. And COVID-19, the same thing. You can have COVID-19 without kidney disease, but if you have proteinuria, it can either be associated with acute kidney injury, which is a tubular issue, or it can be associated with the collapsing phenotype. And what we saw in our study of individuals with collapsing glomerulopathy, without collapsing glomerulopathy, and we're not showing it, but without either, we saw that when we looked for cathepsin C staining in the capsule in the parietal epithelial cell on Bowman space, it always stained. When we looked at the number of glomeruli that had activated PECs, in all of these conditions, we had from 50 to 70% activated PECs. But when we looked at the tuft staining area for cathepsin C, we saw that in the individuals without collapsing glomerulopathy, the percentage area was only 3%, whereas those with collapsing glomerulopathy had 12 to 13% stained area. The fraction of glomeruli staining for cathepsin C in the collapsing FSGS negative patients was 5% versus 38 to 52%. Similar findings for annexin A3. So it seems like with collapsing glomerulopathy, there's a common cell phenotype, a parietal epithelial cell that migrates into the tuft and stays hyperactive and contributes to the sclerosis. That's the hypothesis. So I'm going to jump ahead and in the last 3 minutes cover a few slides. So we're interested in improving our approach. And so you need to improve your approach because you want to improve your statistics. So we know we have variation of phenotype in a particular biopsy, whatever biopsy it is. So if you're doing a liver biopsy and you've got some sort of fibrosis or steatosis, it's focal, it's not global, right? So, but if you're studying it in an animal, you're cutting a chunk of tissue out and you're pulverizing the whole tissue, so you're losing your sensitivity to specificity. So we do have variation, similar variation in patient biopsies. So our goal is to improve the method so we improve our statistics. Oftentimes we have limited sample input, so we need to improve our methodology so we're more sensitive to lower input amount. And lastly, and what I think is going to be the greatest benefit possibly to being having significant contributions is the ability to use archive tissue. So this is an example of one study that we're conducting with people in Columbus, Ohio, and in WashU in St. Louis. And what we're doing here is we're looking at a particular type of glomerular disease called crescentic GN. I'm not going to go into why that's significant, but we're networking with pathologists. So Michelle Barati has the ability to network in with a group of pathologists that have digital imagery, and they're able to annotate the images and transmit that information for her to use to cut glomeruli out of serial sections. And then those glomeruli we're analyzing in a fashion where we're aggregating all the sections that are specific to one glomeruli. So we're looking at a glomeruli versus another glomeruli in the same biopsy. So we're able to characterize variation within the biopsy per glomerulus. And that's important because some of these glomerulus have a particular disease phenotype with crescentic GN where you have a fibrocellular cap in the urine space. So you don't want to isolate all your glomeruli and smush them together. You want to retain that information. And so this is leading to some pretty phenomenal results already. And so the system she's using is called Viper. I don't know anything about that. She's actually, so they're cutting the tissue sections there. They're first and last for a glomerulus. They're staining with PAS. Then the rest of them, serial sections, are going on these membrane slides that are being shipped here. And she's retrieving a sample and then we're processing. Right, so so this is an example where we took 3 patients, and from each patient we collected a total of 10 glomeruli. 5 glomeruli had a crescentic phenotype, 5 had a normal phenotype. And we compared the variation within the individual and then averaged it across the 3 biopsies. And we're seeing, just with a limited sample input, roughly about, so the plot on the left-hand side is a frequency observation analysis. So you can see that we had 13 crescentic sample inputs and 11 non-crescentic sample inputs. And if we want to be rigorous with our statistical analysis, we should just be analyzing proteins that are observed in all samples. If you have a missing value, then you start having to decide on a method to impute a missing value. So we had roughly 822 to 1230 values that were consistent across all data sets. And so that allows us to perform our volcanograph or our downstream informatics to understand the pathways that may be activated here. So this is a volcano plot represented for just the top features here, the top 1230 features, where the missing values are over here were imputed by using a limited detection divided by the square root of 2. So what you can see is cathepsin is within this domain. There's a variety of other proteins that are associated with they're nuclear proteins. I'm not going to discuss that. Looking at the data for those 1230 proteins, we pushed it through a protein from a bioinformatics package called gProfiler. It's a free package online out of Belgium, I believe. And it's able to mine the data and look for protein enrichment based upon the Human Protein Atlas, HPA, and it can characterize basically a tissue enrichment for your proteome. And what we saw in our proteome is that our samples were enriched in glomerular cell types, which is beneficial for us because that's what we enriched for. It goes through all the classical types of gene ontology enrichments. What's a little bit nice about this pathway analysis tool is it reports back to you transcription factor and mRNA enrichment, imputed enrichment, which Ingenuity Pathways Analysis can do that for you as well, but that's a paid subscription. So this is leading us to have hypotheses about the formation of the crescent. Right. So in the field of renal pathology and in most pathologic fields, everybody's excited about the use of AI to analyze histologic tissue sections. So the AI software needs to be trained to how to segment your tissue, right? Define structures and then quantify. And so in the field of renal pathology, one of the questions that's been asked and kind of answered is, what's the best stain to use to have reproducible results in the various tissue compartments of the kidney? And so it turns out PAS is the most reproducible method. So what we wanted to ask was, can we use a PAS stain section for our proteomic method, right? So our favorite method, I didn't elaborate on this, but the frozen tissue section didn't give us as rich a data set as the FFPE. So most people want to avoid FFPE because of formalin crosslinks, you think destroy the tissue for proteolysis. That's not true, because that shift base that's formed by the reaction of formaldehyde with the lysine side chain is reversible with temperature. So we routinely heat our samples to 65 degrees to drive reduction of disulfides. And so that process reverses the crosslink. So PAS is a fixed stain. And so the thought has been that's a desert for proteomics, and there's no data there. But the PAS, from a chemist standpoint, the periodic acid oxidizes carbohydrates into aldehydes, and then the shift base is formed between the di and the aldehyde. In a typical proteomic method, we're not analyzing for carbohydrates, we're not analyzing for aldehydes, so any data that would be missed anyway, right? So we performed a study where we looked at capture of individual glomerular sections, not compiled sections, not pooled sections, but individual sections. And we evaluated the comparability of no stain versus H&E versus PAS. And we saw that PAS was equivalent to or superior than the other methods. We evaluated whether or not it was the impact of periodic acid, and periodic acid did not have any type of special benefit. So the importance here is that with a single glomerular section, we're collecting data, and with stained tissue, we're collecting data. So all of those longitudinal studies that were conducted in the 70s, the 80s, and 90s, anything bioreposited potentially could be usable for this type of study. And Michelle's done the specific studies with me to find stained sections from 10 years ago, pop the slide off, and collect samples, and we're analyzing it. And so I'll jump ahead. And this is the last thing. This is with Sanjay and Igor. They wanted to have us determine whether or not we could perform laser capture microdissection proteomics on lesions from innominate arteries in ApoE mice, as well as atrial valves. And so they have their study on benzene exposure in mice. They've done their spatial omics, spatial transcriptomics, CyTOF they're doing now. And what we were able to do for them is Igor was able to provide sections on glass slides to Michelle for 5 versus 5 conditions. Michelle was able to float those sections off the glass slide onto the membrane, retrieve the structures, the innominate artery lesion. We were able to conduct our analysis on this. This data here represents what was found by RNA-seq by Igor, looking at this from the perspective of gene ontology. Downregulated genes were associated with extracellular matrix, molecules associated with elastic fibers, elastic fiber formation, degraded ECM. Things that were upregulated were associated with platelet degranulation. Things associated with ATP synthesis and response to calcium. This is the workflow for the proteomic study. So these are the anomalous arteries on the top, an example of a stained section, the glass slide, sample floated over to the membrane, then the retrieved sample itself. The bottom side are the aortic valves. And this is a little bit of a work in progress because those valve tissues are tough. They're a little bit booger to pulverize, but we collected some nice data on it. This is a gene ontologic analysis of the proteome, and the boxes in red are highlighted to correlate with what the transcriptomic data was highlighted. So there's a direct correlation between gene expression and proteome expression. And so basically, I think a lot of what we're trying to do is to support clinical decision-making, trying to provide an approach, data, information, knowledge that would allow us to differentiate rare unknown etiologies from other types of etiologies with the hope of improving the people's care. So these are all the people involved in the study across the US. I appreciate your time. Sorry, I'm a little long.
I'm just glad you said the words cancer and metastasis.
I'm meeting with your people on Friday for the biorepository.
All right.
Because kidney cancer is a prime activity for this method. So, we do have ongoing activities where we're trying to develop a single cell approach. And so when we get that enabled, everybody that's in here that does anything with inflammatory cells, immune cells, I think you're going to be kind of the first people that would benefit from that. For next down the line is spatial omics, multi-omics with a multi-source. But we've got a really nice workflow established for laser capture, even if you're doing stain stuff, 3-micron sections. When they were clearing out the tower on the 10th floor, there were just all these slide boxes of mouse brain sections stained. And it was all I could do to not pile up stuff. Because Michelle would have killed me. It's easy. All this stuff is really easy because I don't have to do it.
So with collapsing, I know probably because of the talk, you couldn't go into this too much, but there's definitely an association with the APOL1 experience.
Yeah.
Did you have any data on like the APOL1 status of any of the biopsies?
I don't have any information on the APOL1 status. We have received antibodies that were developed by Genentech that are optimal for immunofluorescence studies for lipoprotein L1. And we have looked at that to determine whether or not that co-localizes with our markers for the parietal epithelial cells. And I don't think it does. I do think it correlates with podocyte localization.
Yeah. So that was that goes with my next question, which is, there have been a lot of hypotheses about FSGS and whether collapsing just represents the severity of podocyte injury that then brings in the parietal epithelial cells to try to replace the podocytes that have been lost. So maybe it's more of the degree of injury. And my next question was, obviously there's new data that APOL1 variants increase the risk of a patient having podocyte injury, but is there any role in parietal epithelial function, cell function? So that was.
So our plan is to, you can take glomeruli that are microfiltered and you can grow out, if you still have the capsule on, you can grow out the PEX. And so, I know you and Dave Powell, y'all have done some cultured glomerular cell and have experience with treating those cells ectopically. We kind of want to do the same thing. want to evaluate, and some of you guys in cancer have done these things where you extract ECM from tissue and plate it out. So that's the goal for us, is from a patient standpoint, extract ECM, plate it out, and see if there is a interaction dependent upon the ECM with the various epithelial cells out of the glomerulus. So the three proteins that I highlighted, cathepsin C, cathepsin B, and annexin A3. Cathepsin C is actually it's a protease, it's a thioprotease. It's involved in COVID-19 infection. And there's a major clinical trial initiated out of Great Britain dealing with cathepsin C inhibitors and COVID complications. But they haven't reported yet on any type of benefit for renal
Yeah,
complications.
I
So,
think the main case series on COVID-associated collapsing FSGS show that the vast majority of the patients had the April risk variants, so,
so with our patients, we do know their race,
okay.
right? And so there were Black and white people that had the collapsing and the COVID. I don't know why. And we certainly don't have a large sample set. And when Jessica Hatta went to Harvard, that kind of ended my hookup.
Is there a question on the chat board?
Dr. Williams.
It's a comment.
It's a comment.
Only a comment. Just really spectacular, and glad
I
that
think
you're.
that says a research budget of $1 million.
There you go. Okay. But you're doing very well with what you have, clearly. But it's so important to be able to use these techniques in other areas. And I'll be coming to you with my nutrition studies, trying to see how we can change proteomics and lower cardiovascular risk.
Absolutely.
Thanks.
All right.